Welcome to this Bioanalysis Zone interview with Dr. Roger Hayes. Dr. Hayes is the Senior Vice President, DMPK at MPI Research. Before joining the company in 2011, Dr. Hayes held numerous leadership positions in the global life sciences industry and academia, leading teams in the development of state-of-the-art bioanalytical and analytical techniques, including mass spectrometry, chromatography, and automation in bringing medical and chemical products to market. Today we will be speaking to Roger about his work on microsampling and discuss recent developments within the field, including the demand for microsampling, benefits and challenges of the techniques, and what the future holds for microsampling. Hello, Roger.
Hello. Hello, Aya.
Thank you for joining us today. We'll proceed with our first question. Could you provide a brief summary of your career to date?
Well, my career has gone from leaps and bounds from large pharma to multiple mergers and acquisitions to now going into the CRO field, which is where seems to be the majority of the work has occurred. So I spent 20 years in large pharma, and I just had my fifth anniversary with MPI Research and the contract research organisation. So a lot of experience in drug development, and then now more in the routine drug development for other sponsors.
Great. Could you tell us more about your current work?
Yes, I have the fortune of managing an organisation that supports predominantly non-clinical bioanalysis, drug metabolism, PK. So that includes management of an ADME group doing drug metabolism, biodistribution, bioanalytical, both large and small molecule. The bioanalysis goes from discovery through non-clinical, GLP toxicology, and then onto the clinic. We've done a lot more work now, if you want to talk about more current, in the phase 3 clinical space, both large and small molecule. We also have I have responsibility for analytical, which is supporting non-clinical toxicology, formulation support. We also have some niche things that I have to manage, being the Translational Imaging Center. So that's a unique modality. So it's a span the whole gamut of DMPK from discovery through to phase 1 and then into phase 3 for the bioanalysis. So a lot of the bits and pieces that go into drug development.
Thank you. What does microsampling mean to you in a sentence?
Well, very simply and very practically, a microsample. So for me, it's a small aliquot that's for bioanalysis that is less than 50 microliters. That aliquot could be serum, plasma, blood, or even CSF.
So what began your interest in microsampling?
Well, it actually occurred in parallel with the need to improve assay sensitivity. So as we had more and more potent drugs, we had to improve detection limits using normal volumes of sample. And then, of course, the byproduct is as we improved sensitivity, it meant that for, let's call them normal drugs with reasonable detection levels, we'd use less sample. It really kicked off for me in the 2000s when we were doing a lot of discovery PK screening. And these are new molecular entities that our medicinal chemists would synthesize, and they were very keen on understanding the pharmacokinetics. And so that was the amounts of test article were, milligrams of material. So you didn't have a lot of test article to dose a lot of animals, and that then led to finding techniques for even smaller volumes to reduce the amount of test article being dosed. They were also trying to come up with good quality data, and so the serial sampling was the ultimate for data quality. And so with those two component, small amount of test article with high quality data in a as fast as possible scenario led to just techniques and a workflow that relied on small volumes of sample.
Great. So how has the demand for microsampling increased in recent years?
Well, I'd argue it was always there. Anytime that you can do a bioanalysis with as little as possible sample and push detection limits, that's always been there. I think the industry and the buzzword of microsampling has sort of been confused a little bit because it's been like a dried sample. The dried blood spots seem to be the real push, or at least microsampling coming into the vernacular for bioanalysis. And really, I think the dried blood spot had its place with simplifying the sample shipments, the opportunity for home-based sampling, pediatric trials and all of those kind of things. But microsamples have always been there. What has happened is instead of doing, half mil, plasma aliquots for bioanalysis and fairly extensive concentration workflows to get the sensitivity we need, as the instruments got more and more sensitive, half mil became, 50 microliters, and now it's 5 microliters. So really the microsample has always been there for me, and as I said, in the 2000s, that's really where mass spec bioanalysis took off. And now with the biologics, there's these narrow dynamic ranges, incredibly sensitive detectors. For us, 2 microliters is the top end, and we're doing nanoliters on platforms like the GIS workstations. So it's always been there. The technology's just improved, and it's just got easier.
So following on from that, what do you think are the main benefits of microsampling?
Well, if I didn't say the 3 R's, I'd be frowned upon. So really the replacement, refinement, reduction of animals, that's the key. But there's a quality that's associated with that. The better you can do serial sampling, the better the quality of the data coming out of what you're analyzing. When you're using fewer animals, and this is more strictly in the discovery and non-clinical space, anytime you have fewer animals on study, everybody's happier. Your IACUC is more pleased, but there's a cost reduction as well, and so you get the quality with a sort of a cheaper study. The serial sample, as I mentioned before, is the gold standard for doing pharmacokinetic analysis. There's less variability when you're taking blood out of the same animal. On the in-life side, it's actually faster. You don't have to wait as long to get that sample. So for a single time point, you can only take about 10% of the blood volume, and if you're taking most of the blood volume at a single point, and it takes a while to actually bleed the animal. With these microsamples, and certainly with the volumetrically accurate devices that are out there, the VAMs, those are 10 microliters, and it's much, much faster, causes less stress on the animals. Certainly the accuracy there with these volumetrically accurate devices, and that just improves the overall quality. The PK timing, trying to get a blood sample, 15 minutes, 30 minutes, 45 minutes, when you're trying to warm an animal up, to get the normal, let's call it normal approach to bleeding, that oftentimes leads to problems if an animal squirms or something like that, or you just can't get the blood flow. So it just improves the overall quality of the data, less animal stress, all of those kind of things.
Great. So in contrast, what do you think are the biggest challenge or the biggest limitation microsampling faces today?
Well, right out of the gate, regulatory approval. Certainly within the US, the FDA are taking a very conservative approach. for the regulated studies, and this is both in the GLP toxicology as well as the clinical space, you need to do these complementary assays, both liquid, and I'm really talking here again about the dry blood spot. Switching over to small liquid samples, there's no real issue with it. In fact, almost 100% of our methods in bioanalysis for the tox work, they're all microsamples. It's just the understanding that 25 microliters is more than adequate, and that is actually a microsample. That's absolutely routine for us. But when it comes to the overall perception, it is a regulatory approval of these dried blood spots, which has slowed the real the explosion in that technology. But there's ways around it. Again, these VAM devices is, I think, the way that it will become very routine and sort of sidestep that liquid collection. Companies are our sponsors are very conservative, and if you say that you're doing something new or let's say out of the ordinary, they get very nervous. They say, well, why don't you do that for some other company? But I want this to be rock solid. I'm spending $1 million on a tox study. I don't want any doubts. And so there's that conservative in the U.S., but when we get European sponsors, there's a little bit more amenable to focus on that reduction, the 3 R's. But for what we've seen, it is a conservatism that, are you sure it's going to work? Now, we've obviously done all of our due diligence within our laboratories, and as I said, we've been doing small volumes of samples for 20 years. There's no real issue, but it's the dry blood spot that gets all the press. The issues with hematocrit and things like that creates doubt in people's minds when you say microsamples. Now, we just say we're doing small volumes, and it avoids that issue.
Great. So I think, you sort of answered the next question, but you mentioned microsampling has always been around, and there's still some conservatism within people in the industry in accepting it. Do you think there are any other reasons that's preventing laboratories or researchers from applying microsampling, or is just this kind of the main reason, only conservatism and the regulatory side of things?
I think it is the regulatory approval within the U.S. I think Europe, that's not really there. I think the regulators are much more amenable to accepting it. And I'm really talking about dried matrices, and that's where I think those opportunities for clinical studies where you can do home-based sampling. Or pediatric training, it's just small volume things, that will force the situation where people will have to do it that way. And as that body of data builds, and there certainly is a lot of large pharma that are doing a lot of work, and they are pioneers, regulators will get comfortable. And that will take a little bit of time. I think within the US, by about 2020, there'll be enough data for the regulators to say, this is really routine. But certainly in my space, with a lot of our work in the non-clinical, GLP tox, it's just advancing as instrument sensitivity improves. As we're doing more and more biologics, just the assay formats, we're already in the microsampling paradigm. So that's been accepted for many, many years, but it's the vocabulary around microsampling, and that's being associated with the dried matrices may really be what's slowing down the full implementation. That will disappear. As people get more and more educated about what a microsampling is, there's no mystery around it. It'll get comfortable, and then it'll become as routine in the clinical space as it is in the non-clinical.
Great. So, what surprises have you encountered in the field so far?
Ooh, surprises. Well, that's it really took off with the dry blood spot. Everybody was really gung-ho. Some of the pioneering work for GSK, we started to do it. When it came to a screeching halt, when a regulator stands up and says, well, we really don't feel comfortable about the quality of the data. So that was a very abrupt change in momentum, and that perhaps for me was the biggest surprise, because we thought we had things squared away with the regulators. But that's probably the main surprise. To me, it's this baggage that we're carrying around with the dry blood spot, and the liquid microsamples are working just great.
Great. So where do you see the field in the next 5 to 10 years?
Right, so the conservatism will disappear. I think it becomes absolutely routine. Instrument sensitivity will continue to push the vendors for more and more sensitive equipment. As I said before, in parallel, that will reduce the amount of volume that is needed to do the bioanalysis. We'll go from 96 to 384 well plates, just a higher volume. Regulators will start approving NDAs, so these are the new drug applications, or the BLAs and the biologics. The concern will vanish. I think that will happen more in the 5 years. Instrument sensitivity will continue to improve. I would think every couple of years we may see I don't know if we'll see the log changes in sensitivity. That just makes it even easier. So I think the tools that become available for handling small volumes, the accuracy of the sampling devices, just the workflows, the automation. It is not trivial to do 384 well plates. We do them. It is not yet routine. 5 years from now, we would laugh at it and say, well, when are we going to move up to the next platform? 384 is easy. So I think just the ease and the workflow, the accuracy of how we go about doing the bioanalysis will improve. And then with that, the regulators will become very comfortable, and it will just become part of the normal course of business.
Great. So if you could give a piece of advice to fellow colleagues looking to implement microsampling, what would you say to them?
Well, first you've got to assure them that the study will be valid. There's been enough noise out there about regulators not approving it, and we have to do all of this extra work and so forth. I think the first piece of advice is just we've been doing it for 20 years. It's just the dry blood spot which is the issue at the moment, but even that is being resolved. So bottom line, your study is going to be valid. It won't cost more. In fact, it'll actually cost less, and that's always a good thing when you're trying to keep your budgets in check. So certainly as we have these discussions with sponsors and saying, look, it's actually not a bad thing. The data's valid. It'll cost you less. It's not more difficult. Again, we've been doing it for a long time, and these devices for volumetric absorption are making it even easier, and it's easier on the animals. So at the end of the day, you still need a champion within the organization to drive it, to be that resource to say, well, can we do this? Do we really have to? You've got to get over that activation barrier, and that's true of any, even incremental changes. You still need someone to push it. Let's give it a go. Let's do the internal due diligence to make sure we can actually, take a 10 microliter sample out of a mouse. You need that. So that internal champion's perhaps it's still there. And always you have to communicate. You've got to communicate with your sponsors saying, the studies are going along well, the data quality is there, and then just continue to reinforce those advantages. So that's, really the advice you give to anybody about any new, I can't even call it a new technique, but any workflow. It does work, it's routine, there's nothing to be scared about, there's no need for conservatism, it works.
Great. Do you have any additional comments to add, or?
No, I think at this time, I think we've covered most things, Ian. It is an exciting time to be doing bioanalysis. I know, in 20 years, going from, half mil to a mil volumes to now thinking that 2 microliters is routine, that's exciting. I'd like to see what the next 10 years looks like. It's almost unimaginable to think that we've dropped, multiple logs in volume, and the sensitivities, picograms quite routine. We used to think micrograms was tough. So it's an exciting time.
Great. Well, it was great speaking to you, Roger. Thank you for your time, and goodbye.
All right, Ian. Okay, thank you.